Hello, this is Mistaken Forward, and we're going to keep working on transcription and translation. This time we're going to look at how everything is regulated. Now we're going to start with bacteria, and what's going to be amazing is, remember I told you that the eukaryotic systems are more complex. Well, when you're going to see the bacteria, you're going to think that's not true, because the bacteria seems pretty complex. Now, I will tell you, though, if you can remember a couple of if-then statements back when you were in biology, if-then, the proofs, if you can remember that, then you can work your way through the operons. It's really helpful to understand why they work, and then that helps you remember how they work. All right, so we're in bacteria, and bacteria are going to have one of the most basic types of regulatory systems, and they're called operons. Now, operons, you may have heard it just a little bit in biology class, so we're going to go a little bit deeper. Operons are a series of genes related to the same metabolic function. Now, you might remember in my previous video, I talked about a teddy bear factory. So imagine the teddy bear assembly line. Their entire job is to make teddy bears. So in the same way, an operon has genes related to the same job. They're making something or breaking something down, but they're all doing something about that. So if we want them to turn on at the same time, or if we don't want them, we want them to turn off at the same time. So that's the idea behind an operon. We have a series of genes, they're all related in some way, and we have a switch that will switch all of them off or all of them on. So we have the same switch. Now the switch is called an operator. And then the promoter we've already talked about. The promoter is where the RNA polymerase starts everything. So let's take a look at it. You have this picture, and an operon has similar features. They are going to have the promoter. So here's our promoter region. And we have the switch. So this is the operator. And then we have the genes for making whatever it is we're making, or genes for breaking down. In this case, we're going to make something. This is called the trip operon. And the trip operon is for making tryptophan. Now, tryptophan is an amino acid. Now, the upstream elements, we have an upstream regulator gene. Now, the upstream regulator gene is going to be making a repressor protein. So this is called TRPR for regulator, and it's going to make, well, you're going to get the mRNA for it, and then you're going to make this repressor protein. Now, this repressor protein is going to have a very specific shape. And I'm hoping you can start to put together this idea that this repressor protein will bind to the operator. It has the shape. Now, as it stands in this picture, it can't bind. Notice this shape will not fit in the yellow shape. So let's figure out why this works. So over here, well, first of all, actually, let's take a step back. In this picture, notice there is nothing blocking the operator. Nothing is there. So notice the arrow. The arrow is indicating that RNA polymerase is going to happily continue, and then you're going to actually make those genes. Now, these genes are for making tryptophan. So let's think about the conditions that would work. The conditions where this is going to be on is if tryptophan is absent. So let's think this through logically. If tryptophan is absent, then we need to make it. So there's our if-then statement. If tryptophan is absent, then we make trip. Now, how do we make trip? We make trip from other amino acids. So you might recall that amino acids have this very basic nitrogen, carbon, carbon shape. Now, I'm not quite interested in worrying about like the specifics here, but you want to think about the idea that if we want tryptophan, then we want to take one amino acid with one R group, and we want to convert it to our amino acid. Whoops, that should be carbon. Sorry about that. Um, the other amino acid with the other R group, because remember, that's the only thing that differs from one amino acid to the next is the R group. So we're going to make this tryptophan from other amino acids by switching and converting the R groups. But that doesn't happen spontaneously. We need we need enzymes. We need enzymes that do that. Well, lo and behold, where are we getting these enzymes from? This operon. We're going to take the genes, we're going to transcribe them, and we're going to get enzymes that can convert other amino acids to tryptophan. Well, that's all well and good. So notice here we have the reason why this works. If tryp is absent, then we need to make tryptophan. So therefore, we want this operon to be on. So this is turned on. So there is our big take-home message. So that is if we don't have tryptophan, we make everything. That's okay. Now, what if we do have tryptophan? Now, if you're a bacteria, this is going to be dependent upon your environment. So let's say you're a bacteria in us and you're not getting tryptophan. Well, you're going to turn this operon on, or at least keep it on. Now, if tryptophan is present, so again, if you're a bacteria in us and we're eating plenty of foods that have tryptophan, so if tryptophan is present, then we actually want to shut down the operon because we have plenty of tryptophan. Now, the way this works, this is actually pretty clever. This is pretty neat. So the way this works is we have our gene, the regulator gene, the TRPR. So here's our TRPR mRNA, and then we make our repressor protein. Now, what we need is the tryptophan. Tryptophan is going to come along, bind this repressor protein, and ding, 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 this is the big thing. It changes the shape of the repressor protein. And then that's going to bind to the operator, and it blocks our RNA polymerase from continuing. So again, the only reason this works is because we have plenty of tryptophan to bind the repressor protein, and then that changes the shape, and then that binds to the the operator, and then that blocks it. And then lo and behold, we're saving energy. We're saving energy because we don't need to make all of these enzymes to make tryptophan when we already have it. So that's the trip operon. Now, notice the similarities that we've got the regulatory gene. We've got our that's upstream. It's outside of the operon. And we'll talk about why that has to happen. And then we have another one called the lac operon. Now, the lac operon is going to be for lactose. Now, just a quick reminder, lactose, let me zoom in on this so you can see it. Lactose is a sugar. It's a disaccharide. Most organisms can't use lactose by itself. You need to break it down into monosaccharides. So we need enzymes. Enzymes are proteins. Proteins are governed by genes. Those are the instructions to make the enzymes. So again, we've got ourselves a couple of if-then statements. So here's our if-then statement. If lactose is absent, so think through it logically. If we don't have lactose, then why make the enzymes to break down lactose? We don't have it. So here's our lac operon. We've got our, and really all we need to look at is that repressor protein, or in this case an inducer protein, but the regulatory protein. All we need to look at is the protein that regulates it, and then the operator to see kind of how everything is working. So if lactose is absent, then we need to stop the production. So this is turned off. Now, when I was learning this back in undergrad and then again in grad school, these little if-then statements helped me a lot because it made me see why it was working. If we don't have lactose, then the last thing we need to make is the enzyme that breaks down lactose. So we want to turn this thing off. So here we have the way it turns off. Well, we have this inducer protein that has a specific shape, and lo and behold, it's now binding to the operator region and blocking RNA polymerase. And again, this is an upstream gene called LAC-I for the inducer. Now, what if we do have lactose? Well, if we do have lactose, well, then we do want to make the enzymes. Now, it's not just enzymes. There's also a transporter protein. There's also a protein that carries the lactose. So we want the operon on. So let's take a look at this. Let me zoom in. This is pretty tiny. So here we have our promoter region, our operator, and then the genes to break it down, break lactose down. Now, in this case, think about what's driving this. If we have lactose, so if we have lactose, then suddenly the cell is going to signal that we need to get these enzymes and proteins responsible for breaking it down. So we're going to get our lactose or allolactose, which is just an isomer. It's going to bind to our regulatory gene. It changes shape. And now notice it can't bind, and it's going to latch on. It's not going to be able to latch on to the operator. And so therefore our RNA polymerase is going to continue down, and you're going to transcribe the mRNA. You're going to then make the enzymes. Beta-galactosidase, which is the enzyme, permease, which allows lactose inside, and then it's going to increase the permeability, and then an enzyme called transacetylase, which helps carry the lactose. And so what we have is a system that makes these proteins when we need them. So here's our kind of if-then statement. Lactose disaccharide need to break down into monosaccharide, so we need an enzyme to do this. Now, a lot of students will then ask me, well, what if I am lactose intolerant? Well, a couple of things you need to remember. This is in prokaryotes, so we see this in bacteria. So this is not operating in our cells. But this can have an effect for the bacteria that's inside of our intestines. If we're lactose intolerant, then we're probably not drinking milk, or certainly not a lot, or we're drinking lactose-free milk. And then the bacteria is not getting lactose. And then there's no need to make the enzyme. So just a clarification on that. The bacteria have the operon, not our intestine cells. But that's a good, it shows that you're thinking if you're getting that. Now, so that's your introduction to operons, which are prokaryotic systems, although we have found a few in some worms and some yeast. We have found some eukaryotic operons, but by far they're much more well known in bacteria. Well, hope this helps. We're going to do some practice for this. Have a nice day.